All right, we're coming down to the last fourth type of tissue found within a human body. And obviously, after epithelia, connective tissue, muscle tissue, the only remaining part is to say a few words about the nervous tissue. So we have here just one slide to use as a starting point for our discussion, but I'm sure that most of you would immediately understand that kind of versatility that nervous system and nervous tissue in general would have to offer to us. So in this first part, we will just address the basic concepts of a nervous tissue without going into action potential physiology, as that part will be explained in a form of separate recording, and it would be clearly titled Action Potential of a Nervous Tissue. For now, we just have to learn a little bit about basic ingredients, components that assemble the nervous tissue, and of course, the cell, which is unavoidable, is very uniquely present in nervous tissue only, and it is the neuron. We call it either a nerve cell or nerve fiber, again, referring to sometimes excessive length of one of its projections that could go past 10, 15 centimeters length or inches. And for now, we can see here that somewhat unusual shape of a cellular body is observed under this photomicrograph. What dominates the picture is quite a large cellular body with a nucleus. And keep in mind that our neurons also have exited cellular cycle and they are in a G0 phase, practically preventing them from continuously multiplying an increasing number of neurons. But there isn't much to worry about. Basically, in the process, when our embryonic bodies and fetal bodies were properly populated, Mother Nature gave us a substantial number of neurons that would be, in most cases, quite sufficient to carry our regular activities without any kind of negative interference of nervous system on a day by day for whatever length of our life is going to be. So within the cellular body, other than nucleus, of course, we're to find all other different cellular organelles, as it has been described previously in a section about cell in general. But what makes neurons being unique amongst any other form of cells is that they have a number of different projections that are coming directly from the cellular body. Multiple projections that are seen on this photograph are called dendrites, and dendrites are matched with just one single projection that is called axon. Makes sense to address immediately what is the role of dendrites versus what's the role of an axon. Dendrites are so-called receiving ends of a nerve cell. The language they use to communicate between themselves is the electricity, action potential, which could be passed from one cell to another, from one neuron to another one, through the synapse connection, which is functional, not physical, but rather functional connection, because between two neurons, there is a small, tiny gap, so-called the synaptic cleft. But regardless of that, electrical signals, once they are generated, they would be moving from a periphery of a cell through the dendrite until it reaches the cellular body. All dendrites will have the same role. They would conduct electrical impulses or electrical charges from periphery into cellular body. So based on what kind of charge the dendrites have been receiving, some of them could be stimulatory, exciting, some of them could be inhibitory, inhibiting signals. Basically, the sum of all stimulus related and all inhibition related signals will be processed by a cell. And on this slightly wider part of the cellular body, which I'm marking now with the arrow that we call axon hillock, it is going to be determined the outcome. Usually, the sum of charges will come up with a need for this particular neuron to generate its own action potential. And when it happens, the newly formed action potential will be sent along the length of an axon. But in this case, it is going to be sent peripherally, away from the cellular body. So why would this be needed? Well, signal, electrical in nature, is going to reach the end of the axon at the axonal bulb, and depending with which cell this particular nerve is creating a synapse, it could be muscle cell, so the nerve would stimulate muscle to do contraction, or it could be a gland. Still, the gland needs to be stimulated to keep producing and releasing its product. So in both cases, we're seeing nervous system as being one of the key elements that regulates many other functions of the body. Therefore, this model of a neuron with multiple dendrites and then a single axon is something that has to be carved in stone and be part of our future studies of the nervous system and nervous tissue in general. Neurons are quite sensitive, and just being on their own, they would likely be under a very negative influence of a number of different events that our bodies will have to go through the life. Therefore, a nervous system, central nervous system in particular, the brain and the spinal cord, will have plenty of additional supporting cells that would be there to practically nourish and protect neurons, enabling them to do their function in the best possible way. These cells could be astrocytes, could be oligodendrocytes. These cells are going to be described and discussed later on when we do anatomy of the central nervous system, anatomy and physiology. But for now, we are moving on to cover some other details interesting for us from point of view what is the rest of the nervous system doing. So let me just switch to a different slideshow and to use this picture, just identifying a variety of different components that are part of the nervous system. So the best way for us to start would be to take a look and to say, let's identify and let's firmly define what is the central nervous system. Of course, it is composed of brain being housed within the cranial vault inside our head, and the spinal cord, which continues from opening on the foramen magnum, occipital bone, or from a first cervical vertebra, all the way through the length of the spinal column, spinal cord terminating in average adult person between first and second lumbar vertebrae. Basically, that is what makes the central nervous system. Peripheral nervous system certainly comes immediately as structure that we have to address. Peripheral nervous system is composed of nerves, and there is a total of 12 cranial nerves. What do we do to call them cranial nerves? These are nerves that originate directly from the surface of the brain, and that's why they compose a unit on their own, 12 pairs, and most of them, with exception perhaps of vagus nerve, would be limited with their action to the head, face, and partly to the neck. I'm again using some common denominator. This is not absolutely accurate, but I just want to give you some form of orientation. Cranial nerves would be those ones that will facilitate our special senses of smell, vision, hearing, taste. Some of them will influence our muscles of facial expression, so we can clearly project ourselves as being excited, being happy, being sad, being concerned, or any other facial expression that we generate. Those nerves will also help us move our tongue. Those nerves will help us properly chew the food which we place inside the oral cavity, and perhaps with the exception of few nerves that are going outside of the head and neck, most of their activity is limited to it. Cranial nerve number 10 is the vagus nerve, and as such is going to be extremely important component of autonomic nervous system. So that's why I need to give you some kind of rough idea rather than to try to follow everything up all the way with all the accurate details. Peripheral nervous system on the top of 12 pairs of cranial nerves is also going to be enriched with additional 31 pairs of spinal nerves. Those nerves originate from the surface of the spinal cord, and there should be 31 of them. Nerves that are arising from the spinal cord are generally given names such as cervical nerves if they originate from the neck part and in between cervical vertebrae. Then thoracic nerves are those ones that are originating from the thoracic region of spinal cord, and then we have lumbar nerves, and then we have sacral nerves, and the last 31st pair is actually the last pair, which is called coccygeal nerve, or coccygeal nerves, because we have two of them, one going to the left, one going to the right. You can also use the idea of peripheral nerves that would be sent further down along the trunk, along the upper or lower limbs. And of course, then we have to see where they really end. If they keep going through our limbs, they would stimulate muscles. Muscles will do the voluntary contraction, but also peripheral nervous system ends up supplying through its sensory component variety of different receptors that are scattered throughout our skin. So when we touch something, we receive immediately a bit of information about what has been touched. Is it something sharp? Is it something rounded? Is it something warm? Is it something cold? And of course, variety of different shapes and textures is what we learn through the lifetime to recognize just through the touch. And we use it to successfully orient ourselves towards the rest of the space that we're in. Interestingly enough, regardless of how rich in innervation the external surface of the body is, you and I will find out a relative lack of same type of innervation for our internal organs. But of course, they're also innervated. However, innervation is based on the autonomic nervous system, which is made of sympathetic and parasympathetic divisions. And of course, sympathetics and parasympathetics, we do not influence these two parts of autonomic nervous system voluntarily. Hence the name autonomic nervous system, the one which will be able to regulate function of a variety of different organs and organ systems, as illustrated here with the presence of all of these tiny yellow dots that are seen within the wall of a small intestine. Small intestine is known as the enteros, therefore enteric plexuses, bundle and network of nerves that are seen throughout the walls of the small intestine to regulate movement of chyme through intestinal space, and also to facilitate and modify our absorption rate when it's required. Okay, let's take a look at the next slide, which is very schematic, but I really like it as some kind of very easy to follow basics of how the nervous system really operates. We can use a little bit of a computer language if you want, and we can put here in the center, central nervous system, to assume the role of central processing unit as in our computers. It is truly CPU, and we know central nervous system, it's the brain and the spinal cord being put in a very, very center. So unless we turn our computers on and give some work to our CPUs, basically computer becomes just inanimate object sitting on our desks. But once we start typing on it and start using any of the programs or applications, basically, we're creating certain tasks that we expect our computer to successfully complete. Tasks that we normally input into our brain and the spinal cord comes from a variety of different receptors. So you can see that each of the three blue-colored boxes are talking about receptors. We're blessed in a sense that for each type of sensory modality that we need to know, we're given a very special set of receptors capable of responding to that stimulus. So, for example, cones and rods within our eye are responding to light. No other part of the body would be able to detect change in light intensity from bright to dim, just our eyes. Also, our eyes are not good for detection of auditory signals. That's what we have our ears for. So, sensory modality called sound could be only detected by receptors that are part of our ear. Smell, taste, balancing the body, tactile sensation, pain, heat, cold are all modalities for which we must have a uniquely designed receptor capable of detecting that. How about X-rays? Well, we know we don't feel their presence because we don't have receptors. So exposure to prolonged time to X-rays would start damaging the body. Unfortunately, we would be unaware of that because receptors for X-rays simply don't exist. Receptors for spectrum of sunlight also doesn't exist because we don't know whether it's high UV index or low until our skin burns. Only when it burns, we experience sensation of heat telling us that we got burned, but we do not experience high versus low UV radiation based on the receptors. That's why somatic nervous system, SNS, would have a number of different somatic, bodily, and special sensory receptors, practically our tactile signals and special senses. Autonomic nervous system, ANS, also would have sensory receptors in neurons, but in a way less physical presence, so lower numbers than somatic and special receptors are present in the body. That's why receiving any signals from the body internal organs is not quite clear as a sensation. When we use a finger, we touch something and we know exactly it was sharp, it was cold, it was feeling a little wet, or kind of not really like a hard matter. Lots of information becomes collected by simple touch. From the inside, unfortunately, the number of receptors is lower, and also the innervation pattern is a bit less than what we see on the body's surface. Finally, the last part of the nervous system, enteric nervous system, one present within our digestive tube, is also loaded with receptors. And as you can see, somatic, autonomic, and enteric nervous system, all three of them feed into brain and spinal cord, practically giving CPU a task to calculate some kind of activity based on the input which has been generated and collected through different form of receptors. If you ask yourself a question here, why is this lowest box, enteric nervous system, avoiding to send signals always to CNS? Basically, it does maintain a little bit of autonomy, especially when it comes to motility of the digestive tube. That means once we started digesting the food that we consumed, releasing it from the stomach into small and later into large intestine, basically we do not need central nervous system to tell it's time for gut content to be moved further forward for the sake of proper digestion. It is happening automatically, autonomically, and it is the small and large intestine which locally determines the need to contract, to relax, and to move content further along the digestive tube. So that is the exception for which we have here a straight line. Once the CNS have processed the entire input received, it must generate the output. So if you're looking for some kind of hypothetically simplifying way of what is input, what is output, let's put it into this idea. Input appears to be question, and output appears to be the answer. If nobody asks you a question, you would not be tempted to answer a non-existing question. You would remain silent. But if someone asks you, what's your name? You will answer it. If somebody asks you, what time is it? You will reply. So you always have to look for cause consequence. And in this case, we're simply saying brain and spinal cord do not provide answers, they provide output. And output will also include different parts of the body. So most typically what we grasp and we see as the most common thing is involving of our somatic motor neurons. So peripheral nerves having motor fibers that are reaching effector cells. In this case, effector cells are our skeletal muscles that will be properly stimulated to do contraction, to do relaxation as per our intended movement. So here is something that kind of mixes things up. Your autonomic nervous system practically detects that your body is slightly dehydrated. It tells that information to CNS. CNS converts the idea of body is dehydrated into recurring thought of being thirsty. So being thirsty practically requires somatic motor neurons that will make you stand, go to the kitchen, open the cabinet, get the glass, open the tap, pour the glass with the water, drink it, drink one more if you're really thirsty, put it back, go back to what you started doing before feeling that you're thirsty. So that's how it works. And of course, in order to get you close to the kitchen, close to the source of water, close to the glass, basically you have to use a variety of different muscles to effect this simple thing, such as you need to rehydrate the body. Sometimes it is the autonomic nervous system which becomes carrier of our motor output, but this time it is involuntarily carried. You realize you're thirsty and you say, right now, I have to finish this recording. You will not stop doing it just because you temporarily felt that you're thirsty. You can delay it and defer it for next 2 minutes, 5 minutes, 10 minutes, and then go and quench your thirst. But involuntary aspect of autonomic nervous system and its motor divisions, whether it's sympathetic and parasympathetic, will practically waste no time. It will immediately start stimulating its own effector cells in this green colored box, whether they're smooth muscle fibers, whether it's the cardiac muscle or glands. So in this room, while I'm recording this, I forgot to turn the heat off. And as I continue talking, the temperature gradually goes up. I realize it's hot. I start perspirating until I realize, okay, I should turn the heat off. That will become my voluntary action, but involuntarily, I started sweating. Or if something happens right down on the street, some kind of, let's say, car blasts its tire and there is some kind of loud bang, of course, I would do this and my heart likely will start beating a little bit faster because this is a fear of unknown, what has caused this bang. And of course, the autonomic nervous system will like to prepare my body for what's to happen. As we know, old proverbial saying, sympathetic response is flight or fight. And that would be a reason for which my heart was making me ready to run away if the situation requires that kind of activity. Parasympathetics will do the opposite. In this case, as we know, parasympathetics are simply sometimes rest and digest. But more about these proverbial sayings when the time comes. Let's take a look at some input that was generated by GI, digestive tract, being sent to central nervous system, properly processed, and then through the autonomic nervous system, sympathetics and parasympathetics, basically motor neurons that are also involuntarily controlled in enteric nerves, enteric plexuses, will stimulate either smooth muscle cells or glands of the digestive system or endocrine glands of the GI tract to become effectors and to increase their activity in order to meet current demand and to keep body. That's always the ultimate goal, to keep body at the prescribed neat homeostatic levels. Here comes another view to typical cell of the nervous system, the neuron, of course, and this is what we have seen already, cellular body with all organelles, or we can see it in the upper handmade diagram. Multiple dendrites, as we can see here, but also multiple dendrites, as we can see practically arising from the surface of this neuronal body along this red colored line. Of course, dendrites do have their own further branching, so that is called dendritic spine. And through each and every of the ends of the dendrite, whether it's this dot here or this dot here or this one here, those little red dots that I'm adding to this diagram, they could be exposed and interact with another neuron. They could become stimulated, and if they are properly stimulated, they will carry the electrical current into the cellular body for further processing. At the axon hillock, it will be determined if this cell will generate its own signal, action potential, or not. Because if we have same number of inhibitory and stimulatory stimuli, basically they're going to just cancel each other off. However, it is not always the case. Some of the stimuli will prevail, will become dominant, and of course then neuron will generate its own action potential that will be sent along a single process that is called the axon. Axon might branch and send those axon collaterals to the side. That means one nerve cell could really influence hundreds of other effector cells, hundreds of muscle fibers, hundreds of glands, hundreds of smooth muscle fibers, just depending where this neuron is observed. And you can also see here that axon appears to be covered by some kind of strange-looking cells, and these strange-looking cells are called the Schwann cells. Schwann cells are acting as the electrical insulator on the surface of the axon, because it's like a wire. If it carries electrical current anywhere the wire is there, basically the current can dissipate into any other electrical conductor. But by keeping series of Schwann cells covering the surface of axon, we would say that this axon is myelinated. Having a coat of myelin, I'm sorry, it's difficult to write at the same time and to keep talking, but I want to write the word myelin on the screen so you can see it. Myelin acts as the electrical insulator and ensures that speed of electricity running through the axon is accelerated, and the gaps that exist between two Schwann cells, as you can see here, are called nodes of Ranvier. They're going to have a bit of extra role for action potential of the nervous system presentation, so it is good to know that they exist. And finally, when the electrical current arrives at the end of the axon, it is going to practically come to this kind of terminal end where each of these rounded ends at the synaptic end are called synaptic end bulbs, and they would likely contain lots of stored chemical substances that we call the neurotransmitter. So as I'm explaining the very basic things, here is a cell A, first arrow, reaching cell B, but not making the physical direct contact with it. That is our hypothetical idea of what the synapse between two neurons would look like. This gap between A and B is what's called the synaptic cleft. So from these red circles, synaptic and bulb, presynaptic neuron will have to release the chemical substance that diffuses across the synaptic cleft and starts influencing neuron B, being called the postsynaptic. The neurotransmitting substance will induce certain changes, so the neuron B will start building up its own charge, eventually resulting in its own ability to generate a signal, to generate the impulse, to fire it down along its own axon, which finally reaches the muscle or gland and induces its further action. But more about this, definitely we have to do in a nervous system physiology of action potential. Not all the neurons look the same. And on the basis of these three models, you can see that one is called the multipolar. That one is the closest to neuron that we have seen on the previous slide. Then we have neurons that are called bipolar neurons. And then we have neurons that are called the unipolar neurons. So right away, visually, I know that you can see the difference, but it might just be difficult to verbalize it. Multipolar refers to multiple dendrites and, of course, always a single axon. Neuron cannot have more than one axon. So then why is this called bipolar? Well, you can see here, there is just one dendrite. However, one dendrite is having a very extensive dendritic spine, so heavily branching out, and that means if multipolar neuron is commonly found practically anywhere through the CNS, bipolar neurons are likely to be limited with its appearance to our special senses, hearing and vision. Do you see this as a benefit or maybe lack of benefits? It's hard to say, but we know that our eyes have limits. And if within our eyeballs we have about 150 million cells capable of detecting light, that would be equivalent of a camera with 150 million dot pixels capacity. We know more megapixels, better quality, but we do not need to be overburdened by our visual experience. So regardless of these 150 million cells detecting light, color, and changes in the light intensity, basically these signals become summed up, and of course then just one type of current runs through the dendrite, reducing, let's say, our cameras, our eyes, from 150 million pixels to some kind of reasonable 25 or 30 million that we can process. So we have sufficient visual acuity, we come closer to the object we observe, we see it clearly with the details, but we really cannot use our eyes to start seeing submicroscopic structures within a cell. We still need some kind of magnification that facilitates that kind of work. So it's really like very specifically designed to prevent overburdening us with too many of visual stimuli, because brain is not only concerned with what we see, but at the same time it's concerned as what we hear, what we smell, what we taste, what we feel with the rest of the body. So this is, of course, towards our advantage. Certainly bipolar neuron will have just one axon, and this symbol that we have here is really nothing new for physiology, just symbolically represents that length of the axon is way longer than this diagram illustrates. So this is just like ad lib, whatever length of the axon you have to add. Unipolar neuron or pseudo-unipolar. Pseudo-unipolar is probably a better word to use because it means falsely looking as unipolar, which if translated verbatim would mean it's the cell which has just one projection. And in this case, it looks like the cell is only attached to the axon. But we see also this type of cell has extensive dendritic spine. We have quite a long axon, and truly the cellular body appears to be just attached sideways to the axon. But this doesn't exclude presence of dendrites. We still have them, but we have one axon and the body shifted sideways to axon, hence the name, looking like unipolar, but really it is pseudo-unipolar neuron. To avoid idea of just 3 types of neurons, I have to bombard you with extra 2, like Purkinje cell is also neuron, but look at its own dendritic spine, more than what we have seen on the previous slide. Purkinje cells are found within the cerebellum, part of the brain, which has need to control our coordinated movement of multiple parts of the body and multiple muscles. So that's why its dendritic spine has to be able to receive literally thousands of different stimuli all at once. Pyramidal cell is uniquely present in part of the central nervous system, which voluntarily controls movement of muscles. So those pathways, we prefer to call them descending motor, voluntary, or pyramidal pathways, just alluding that they are made of multiple of pyramidal shaped cells. Clearly, that triangular shape of neuronal body is what generated this kind of semi-colloquial name for this type of cell. Process of myelinization is very important because it electrically insulates the axons. And both myelinated and unmyelinated axons exist within our body. Not every single axon is myelinated, but those ones that are myelinated are operating at higher speeds with a greater accuracy. And of course, there are those ones that are not myelinated, where speed doesn't really matter, and it is quite well balanced as a part of the system. For nerves in the peripheral nervous system, it is the Schwann cell, which produces myelin by wrapping itself multiple times around the axon. Length that could be covered by a single Schwann cell is 1 millimeter, so 1/1000th of a meter, or 1/25th of an inch if you're more familiar with imperial system than metrics. This is axon, which appears to be properly myelinated, and this is just a sequence illustrating how the Schwann cell simply wraps itself around it. Please remember, it is formation of a myelin, but only valid as information within the peripheral nervous system. In the central nervous system, myelination will be done with the help of different cell, and these two are beautiful photomicrographs. Look, 5,000 times magnification. This is axon, and this dark ring around it is actually the myelin sheath generated by this cell here. That is the nucleus of a Schwann cell, and the rest of it, it's Schwann cell cytoplasm, but tightness of these multiple layers of myelin simply cannot be seen even with 5,000 times magnification. Once the myelinating process is done in the central nervous system, it will generate 2 different colors of nervous tissue. We're going to call it gray and white matter. Myelin adds to this kind of lighter color appearance, so we can immediately make a conclusion that centrally positioned gray matter is predominantly made of neurons that are not myelinated, or neurons whose axons are not myelinated, because we only get myelin on the surface of the axon. Body doesn't get it. Dendrites cannot be myelinated, just axons. So we know what to expect. This is where we're going to see plenty of neuronal bodies and plenty of dendrites, but not that many axons, because axons are typically myelinated. Where are they? Well, they are seen in the rest of this cross-sectional diagram of the spinal cord. Wherever I put these little X's, basically it points to white matter, which is characteristically recognized with literally thousands of myelinated axons that are making it. In the brain, we have the same situation, gray and white matter, with the difference that the gray matter is now pushed more peripherally to be seen on the surface of the cerebral hemispheres, while the core, the inner aspect of cerebral hemispheres, is still done by white matter. There are structures that are seen deep inside cerebral hemispheres, like these structures that I'm marking with some kind of extra X's. You see that they appear to have a darker discoloration. We sometimes call them either basal ganglia or nuclei, but again, more about this would be in a central nervous system anatomy and physiology, where you're going to get proper idea whereabouts we're seeing it and what is their function. Because this is central nervous system, if Schwann cells are myelinating peripheral nerves, we have to say that it is a cell which is called oligodendrocyte, which is cell within the central nervous system doing myelination of axons. I think that in the next couple of slides, we might have some illustration just to show you what oligodendrocyte would look like. This is astrocyte, this is oligodendrocyte. So cell which looks very much like neuron. However, it's not really a neuron, it's a supporting cell of the central nervous system. And with its multiple projections, as you can see, it is covering part of axons by depositing extra coat of myelin, but this is only the central nervous system. In the peripheral nervous system, role of myelination falls under jurisdiction of Schwann cells. I'm sorry I'm taking you through multiple diagrams that we're going to use anyway in later sections when we address action potential of the nervous system. These diagrams will come quite handy. For now, I want to wrap it up and to say this is what we were able to do in order to cover the fourth remaining part of the tissue, the nervous tissue. And of course, the next topics are going to be functionality of the nervous tissue, aka action potential, functionality of muscle tissue, also known as muscular contraction. And these things will be posted separately, clearly marked. Hope that you would enjoy them, and I hope that you have enjoyed what I've done so far. Anyways, thanks for watching it. Please subscribe to Anatomy, I can say, and Physiology Made Simple by Dr. T on the YouTube channel. Thank you very much. See you soon.